Okay. All right, thank you everyone for coming. I am Chrissie from the Tasmanian Devil Immunology Group here at Menzies Institute, UTAS in Tasmania. And today I'm going to talk about the arms race against DFTD, specifically through the manipulation of antigen presentation pathways. So I'll skip the introduction about DFTD as some of us have already introduced. So basically, there are currently two clonally transmissible cancers, DFT1 and DFT2, that are independent of each other. And, you know, as what the other devil person has mentioned, that, you know, it's almost always fatal. There's a high mortality rate whereby devils typically die within 6 to 12 months of manifestation. And there's currently limited evidence for host resistance or even host response towards this tumor cells. And one of the reasons being, you know, the tumor cells have found a way to escape immune recognition by downregulating MHC. As you can see here, this shows MHC expression on the surface of 3 DFT1 cell lines, and they're downregulated compared to, say, a fibroblast cell in the devil. So fortunately, there's an inflammatory cytokine interferon gamma that's able to restore MHC expression on the cell surface. And so, you know, this provided an opportunity to actually prime the devil immune system with a tumor cell vaccine. Unfortunately, interferon gamma, you know, it's a pleiotropic cytokine with multiple different effects, including, you know, inhibition of cell proliferation and also induction of cell death with prolonged exposures. So this actually complicated, you know, the issue with trying to culture large quantities of tumor cells for vaccination. So in this paper that I published in 2019, I basically used an inducible system to control the expression of interferon gamma and therefore the effects of interferon gamma. So I found that, you know, I could allow the expression of MHC at the start while turning interferon gamma expression to stop the negative side effects of it. However, interferon gamma also has another negative side effect whereby it upregulates this inhibitory checkpoint molecule called PD-L1 that negatively regulates the immune response. And so that would inhibit, you know, the devil immune system from properly or effectively eliminating the tumor cell. So I went on to look at a different gene to upregulate MHC on the surface of DFT cells. So I looked at 2 different genes specifically, NLRC5 and C2TA, and they're both transcriptional co-activators that act on MHC expression at the transcriptional level. So they were found to be the master regulators of MHC class 1 and MHC class 2 expression in humans and mice, but nothing is known about its function in devils and how it can regulate MHC1 and MHC2 expression in DFT cells. So what I did was to introduce this gene into devil facial tumor cells and overexpress it and try to look at what are the function of these genes and whether it can upregulate MHC1 on the cell surface and MHC2. So looking at the first gene, NLRC5, I basically looked at the surface expression of MHC on the cell surface by flow cytometry. And if you can look at this histogram, if there's a shift towards the right, it's just telling you there's increased expression level of beta-2M, which is a subunit of MHC class 1. So if you look at the second row, it shows you wild-type DFT1 cells that do not express MHC. And then with interferon gamma treatment, you can see that MHC is upregulated on the cell surface. And DFT1 BFP is just a mock cell line that has the empty vector with no gene of interest. And this is the NLRC5 overexpressing DFT1 cells. And you can see that MHC1 is definitely upregulated on the cell surface. So the next thing to look at was the second gene, C2TA, and whether and its function, whether it upregulates MHC1 or 2. And if you look at the bottom row, it shows that this gene, C2TA, actually upregulates both MHC class 1 and also MHC class 2 on the cell surface of DFT1 cells. So now that I've shown that, you know, this gene can upregulate MHC1 or 2 on the surface of DFT1 cells, I wanted to investigate how these genes actually upregulate MHC on the cell surface. So the presentation of MHC molecules on the cell surface requires, multiple proteins working together to facilitate the expression, first of all, the synthesis and also the loading of peptides onto MHC-I before it goes up to the surface. And this shows you the pathway of MHC class 1. So I just did RNA sequencing of my cell lines and looked at the transcriptome to see what genes of this pathway is actually upregulated by NLRC5 or C2TA. And if you can focus on the last 3 columns and compare the last 2 columns with DFT1 BFP, that's the negative control, you can see that NLRC5 and C2TA definitely upregulates multiple genes that are involved in MHC class 1 pathway. And the redder it is, it shows you the higher expression level, and the bluer it is, it shows you the lower expression level. And this is just relative expression across the different cell lines and also between the different genes. So looking at MHC class 2 pathway instead, so these are all the important proteins that are involved in MHC2 expression on the cell surface. And you can see that C2TA upregulates multiple MHC class 2 genes, so SAHA-DAA and SAHA-DAB, and also accessory proteins that are involved in, transport and presentation on the cell surface like DMA and CD74. So the next thing to, look at was whether having MHC-I or MHC-II expression, whether it actually enhances DFT cell immunogenicity, whether it actually improves the immune response against double facial tumor cells. So to do this, I took advantage of pre-existing serum antibodies that we found in a few wild devils that had tumor regression. And they also had, antibodies specifically against interferon gamma treated DFT1 cells. Presumably it's against MHC-I, but now that we know that interferon gamma upregulates multiple things on the cell surface, it might not be MHC-I. And this shows, a devil yaka that was, this was discovered by my colleague Camilla. It shows you how, tumors have regressed after 3 months, which is really, really fascinating and amazing. And this devil also had antibodies against devil facial tumor cells that were treated with interferon gamma. So to assess whether MHC is actually the antibody target on DFT cells, the thing to do was to compare how did the antibody response or binding change when MHC was present on the surface and when MHC was absent on the surface. So to do that, I basically created a panel of DFT1 cell lines that had variable MHC expression. So I've got the wild type DFT1 cells that don't have MHC expression on the surface, and I've got interferon gamma treated DFT1 cells with MHC-I upregulated on the cell surface. And I also knocked out beta-2M in the wild type DFT1 cells, and that should render them irreversibly negative for MHC-I expression, as you can see from this antibody binding. And then I had the mock cell line DFT1 BFP that should be MHC-I negative, and the NLRC5 cell line with MHC class 1 that's upregulated on the cell surface. I also knocked out beta-2M in this NLRC5 expressing cell line, and this shows that there is no upregulation of MHC on the surface. So and then looking at the antibody response, so I basically incubated the antibodies with DFT1 wild type cell line. You can see that these cells do not have any target on the cell surface for the antibodies to actually recognize them and bind them. As soon as, I've got interferon gamma treatment on these cells, you can see that the antibodies do bind the cells, telling us that there's something on the cell surface that's a target for these antibodies. And when you knock MHC out of the cell and it's no longer presented on the cell surface, you can see that antibody binding is actually lost. Indicating that we've removed the target on the cell surface. And we see this same pattern with the NLRC5 cell line. With MHC expression, antibody does bind to the cells, and when MHC is lost, you can see that there's loss of antibody binding. So this tells us that MHC is the predominant target of these anti-DFT antibodies, and therefore an important target for humoral response against DFT cells. So I also did this using the C2TA cell lines that express both MHC-I and II to look at how antibodies actually respond to these tumor cells. So you can see that CIITA also upregulates antigenic targets for anti-DFT humoral response. So in the arms race against transmissible cancers by trying to upregulate MHC, I've managed to manipulate MHC-I and MHC class 2 antigen presentation pathways using these two genes. And it's been shown that it could restore DFT cell immunogenicity by upregulating targets for anti-DFT humoral response. And so this just tells us the importance of MHC in anti-tumor immunity and how it actually shapes the immune response against the epithelial tumor cells. And future use would be to exploit this for vaccine development and immunotherapy by selecting specific MHC alleles that are most different between the tumor and host, or even selecting immunogenic MHC bound peptides. So that's all for my talk. Thank you for listening. I'd like to thank my supervisors and the Devil Group and the Tasmania Devils and other researchers who are helping me do this experiment, and also funding bodies. Thank you.